Lab 7, Evolution of Multicellularity. In your group speaker, you should have 2 culture tubes with yeast growing in them, labeled appropriately. You should also have 2 new unlabeled culture tubes with no yeast. Label these appropriately. You should have several supplies at your desk. See the lab manual for details. Starting with one of the previous lab section's culture tubes, swirl the tube by hand to mix. Make sure to pay attention to the labeling. In this case, we are working with a. As seen in Figure 3, extract 5 milliliters of the turbid yeast culture using the glass pipette and pump. Draw up to the 0 mark. Make sure you put it in the appropriately labeled tube, in this case, a. Seal the bottom of the glass pipette with a piece of parafilm, and allow the pipette to stand upright in a glass cylinder or flask for 10 minutes total. Use a timer to keep track of the time. Make sure the media is not leaking out. After the 10 minutes is up, obtain a ruler. Starting at the bottom of the glass pipette, measure the length of the total volume of liquid in the pipette in millimeters. Also, starting at the bottom of the glass pipette, measure the height of the cloudy cell haze in millimeters. Record these data in Table 1. Then, transfer the bottom ½ milliliter to the new culture tube. Make sure to keep track of the labels as you go. Start by looking at the 0 milliliter mark on the pipette and release liquid until it has gone down ½ milliliter. Return the remaining 4½ milliliters to the original tube, making sure to pay attention to the labels as you go. Dispose of your used glass pipette in the glass disposal box. Give your TA the old culture tube for proper disposal. Throw the used parafilm in the trash. Repeat the same process with the other sample.